Even though people are straggling in, I think it's best that we get started so that we don't get behind the 8 ball. Yesterday we were running quite late, and today I'd like for us to finish on time since I know there are a number of you who have to catch airplanes. I want to make one announcement, and I'll make it again. There is an invited luncheon sponsored by GlaxoSmithKline that will be held in the Aberdeen room, which is directly across from here, for invited guests only, but it will be starting at 12 o'clock, which means, unfortunately, that those invitees will miss the poster presentation. The main luncheon, which is at 12:30, which is sponsored by Onyx, will be on time. So now, without further ado, let's head on into our plenary session, and it gives me great pleasure to introduce the first session on cell therapy and to introduce our two chairs. Dr. Spall, who needs no introduction, she's professor and head of the core transplant program at MD Anderson Hospital, and Dr. Asano, who is professor emeritus of the Institute of Medical Science at the University of Tokyo, who will chair this session, and I will turn the program over to them.
Good morning. I welcome you all for myself and Dr. Asano, and we're going to begin with our first talk, Advances in Immunotherapy for Lymphoma and Myeloma by Dr. Larry Kwak from MD Anderson.
Okay. My name is Asano, okay. I'm from Waseda University, okay. I'm working in biochemistry now. Okay, thank you very much. Please, I'm sorry.
Thank you. It's a pleasure to be here, and it's very impressive. Everyone up bright and early, even 7 o'clock on a Saturday morning. I was expecting to see more of my colleagues from America here, but I guess they finally all fell asleep. So in the interest of, in the spirit of transparency, I don't think we're offering CME, but I just wanted to list my relationships with companies. The primary product that I'll be discussing today is originally developed in my laboratory at the National Cancer Institute, has now been taken over by a company called BioVest International, who sponsored the phase 3 trial, which I will summarize for you. Now, I'm going to focus my talk on cancer vaccine therapy, and then at the end show how we integrate that into the adoptive T-cell therapy approaches that you'll hear about in the subsequent talks today. But this is a very exciting time for cancer vaccines. We have now at least 3 or 4 positive controlled phase 3 clinical trials just in the last couple of years. And already we have 2 of these that are already approved products by the FDA, which are listed here. But these include vaccines for prostate cancer, melanoma, and the lymphoma vaccine, which I'll describe to you. Now, the vaccine idea is illustrated on this slide, and it begins with identification and isolation of a tumor-specific antigen, so a target, if you will, on a cancer cell. Isolation of that protein or target, and then appropriate packaging in a delivery system before injection back into the patient as a therapeutic. The therapeutic works indirectly because it doesn't have an anti-cancer effect on its own, but rather it activates the immune system, principally the T-cell immune response, here shown CD4 and CD8 positive T cells that are specific for that target and in theory will activate or will go to the target. We had the privilege over many years now of developing a single target for cancer vaccines, which I'll focus on. This started with preclinical work, again, at the National Cancer Institute in my laboratory, in which we optimized the vaccine delivery, performed phase 1 and 2 clinical trials, which demonstrated molecular remissions, and then finally a phase 3 controlled clinical trial published a couple of years ago in which the vaccine was demonstrated to prolong disease-free survival in patients who were in a minimal disease state. And I'll summarize, these are published, but I'll summarize some of these, just the highlights of this study to illustrate. Now I'm going to apologize in advance for focusing on just one target today, which we term idiotype, but hopefully it'll serve as a model for immunotherapy development in for other hematologic tumors. The idiotype was a target that I was introduced to many years ago by Ron Levy when I was a fellow at Stanford. And Frida Stevenson and Ron Levy actually first described this concept of the variable regions in the immunoglobulin receptor of this surface immunoglobulin on malignant B cells, which can be recognized as antigens by antibodies and more recently from our own work by T cells. So as you can see here, this is a truly tumor-specific target because each receptor, because there's clonal proliferation that gives rise to the tumor will have a receptor of, as shown here, just of a single color, which will distinguish it from all other receptors, normal B cells in the body. So the consequence of having a truly tumor-specific antigen is that this antigen is actually personalized. And it must be made on a customized basis from each patient's tumor because even though you have two patients with follicular lymphoma who look the same under the microscope, the molecular signatures are going to be slightly different. And so this slide illustrates the strategy that we've used over the years to isolate that antigen. It's basically making a human hybridoma to get these tumor cells to secrete the protein into supernatants, where this process can then be of purification can be automated and then used for vaccine production. And the vaccine, this first generation vaccine, actually consists of 3 components. In addition to the idiotype antigen shown here, it is chemically conjugated to a large carrier protein called KLH and has been used in immunology for many years. The purpose of which is that this larger protein makes the smaller protein more visible to the immune system. And then the third component is GM-CSF, which functions as an adjuvant by inducing the migration of dendritic cells to the vaccine site. So the other concept I want to develop with you is that from the beginning, we have always felt that cancer vaccine therapy will work best in a minimal disease state rather than advanced tumors. So the standard phase 1 setting against the multiply relapsed tumors isn't really optimal for cancer vaccine therapy because you need an intact immune system. And so while many cancer vaccines are being developed in the therapeutic setting against advanced tumor, the concept that we really pioneered is that of secondary prevention, that is prevention of relapse. One day, hopefully, it'll be possible to have true prevention vaccines, which have worked so well in infectious diseases. This is the design of the completed phase 3 clinical trial, a randomized controlled trial in which, as I just described, patients were all given uniform induction chemotherapy. The purpose of this chemotherapy was to induce a complete remission or minimal residual disease, and then patients were randomized to receive either the complete vaccine or a control vaccine, which consisted of just the two nonspecific components. And there were 2 prospective efficacy analyses that were planned. The standard intent to treat analysis, which took patients, all patients here, or the modified intent to treat analysis, which I'll focus on, which analyzed just the patients who actually made it to actual vaccination. And these are the Kaplan-Meier disease-free survival curves for the modified intent to treat analysis, in which you can see there is a disease-free survival advantage in favor of the patients who received the complete vaccine shown here. Was statistically significant by a log rank and also confirmed by the Cox proportional hazards model. And these data were with a follow-up of about 5 years with a difference in median disease-free survival of about 14 months between the experimental and control groups. This difference between experimental and control groups was even more dramatic in a subgroup of patients who had just the IgM isotype. So lymphomas can come in 2 types generally, IgM or IgG, and we found in the IgM isotype patients that we've saw a much more dramatic improvement in 4 patients with the vaccine who received the complete vaccine, suggesting this may be a new biomarker to guide future clinical trials. So there are certainly some challenges to the next step that need to be acknowledged here, need to acknowledge that patient accrual was stopped early and the treatment effect was apparent only in the modified intent to treat analysis. Nonetheless, the company is going forward with regulatory approval applications both in the U.S., Europe, and Canada, and I don't think that this is a showstopper. In terms of delivery, there is still a requirement for personalized manufacture and biopsies. So I've heard this product described as scientifically low risk because it's been validated in a phase 3 controlled clinical trial, but high risk from a commercial standpoint. And so maybe it's a little bit ahead of its time in terms of commercialization. And finally, again, optimal treatment requires a sustained complete remission, which is not possible in all patients, although it is possible in the majority of patients. So while the company is going forward with approval, we have lots of work still to do from this trial. One example of this is that we have residual excisional biopsies from all of these patients on the study, which are serving as a source of residual tumor for genomic profiling and biomarker analysis. And my colleagues Satva Nilapu and Eric Davis have secured an NCI R01 grant to do this work, and that is in progress. I think we all acknowledge that additional clinical trials will be needed to combining this vaccine with a Rituxan-containing chemotherapy regimen. I didn't go into too much detail, but the chemotherapy regimen did not include Rituxan. It was a CHOP-like regimen, and part of the reason for that was that the trial was designed before Rituxan became standard of care in the year 2000. But what I'd like to share with you now are our efforts, ongoing efforts in the laboratory to make future improvements in the vaccine product itself and develop second-generation DNA fusion vaccines, which are getting ready now to enter into first-in-human clinical trial at MD Anderson. So the idea behind a second-generation DNA vaccine strategy is to maintain or improve efficacy, but to reduce the manufacturing time. Again, this is to try and begin to address the commercialization barriers of having to make a personalized product. And for protein vaccines, in the phase 3 trial, this took us about 6 months. For DNA vaccines, we can get this down now to about 1 month. And the principle is illustrated on this slide, and is based on the idea of trying to target or harness dendritic cells for cancer vaccine therapy. Now, a very popular approach is shown on this left-hand side of the slide, in which one isolates dendritic cells first from the patient and then loads them with the tumor antigen or in various forms before injection back in. But the approach that we've taken is shown on the right-hand side of this slide, and that is to do all the targeting in vivo, and I'll explain how we do that on this next slide. Basically, it's to make, using molecular biology, to make a DNA fusion construct with between the antigen and shown in red and a chemokine shown in blue. The purpose of the chemokine is that it serves as a ligand to target delivery to chemokine receptors on dendritic cells. So that makes this process of recognition and uptake of the red antigen much more efficient process ultimately for presentation to the immune system. And this is an approach that we've validated in preclinical model systems as shown in those publications. And so we're ready now to take this to a first-in-human clinical trial, and this effort's being led by my colleagues Satva Nilapu and Sheba Thomas, a junior faculty member in our department. And the clinical setting that we've chosen to take this into is, again, this principle of minimal disease. And in this case, it's going to be patients who have the asymptomatic phase of lymphoplasmacytic lymphoma, patients who otherwise would be watched and waited and whose only other therapeutic option is really a toxic chemotherapy. So this is a very appealing, to have a non-toxic vaccine therapy is very appealing to these patients. And we'll be using a standard 3x3 design as shown here with essentially 3 monthly intradermal vaccinations using a needle-free injection device. The final approach that I want to share with you is a collaboration between our group and the stem cell transplant department, principally Drs. Champlin, Muzaffar Kozlobash and Dr. Shapal, in which we're combining adoptive immunotherapy with a vaccine therapy. And what's shown here is the technology developed by Dr. Carl June, in which they activate autologous human T cells with anti-CD3 and anti-CD28 beads as adoptive therapy. This slide shows in a little more detail how Dr. June and the stem cell transplant therapy will be or are activating these T cells from patients before expansion and infusion into the patients. And the hypothesis, this slide was given to me by Dr. Kozlobash, is to combine cancer vaccine therapy, that is, before harvesting those T cells. So, as shown here, the idea is that a cancer vaccine injection into these patients will induce a certain number of tumor-specific T cells in the body, but then with T cell expansion of those antigen-specific T cells, we could take this number up to a hypothetical threshold level that's required for additional clinical efficacy. And so the central hypothesis of this phase 1 clinical trial, again being led by Dr. Kozlobash, is that the vaccine plus vaccine-primed adoptive T cell therapy will result in a robust idiotype-specific T cell and humoral response compared to control vaccine only. So the objective is to test whether infusions of T cells which have been activated by CD3, CD28 beads as described by Dr. June's group, but now are different because they've been primed in vivo with the vaccine first, whether they can mediate a more intense specific immune response. And this is a randomized trial design which has just been activated. I think the first 3 patients have just been enrolled on the study. We expect the study to enroll quickly, and it's supported by our myeloma spore grant. But as shown here, these patients undergo plasmapheresis for vaccine production. They receive the vaccine, priming injections of the vaccine, and then apheresis is performed to obtain the T cells that are expanded by the CD3, CD28 bead technology and infused back into the patient. And then additional vaccine boosters are given after that infusion to further boost the immune response. And so I'll just close with, in a short period of time, I've developed several concepts for you. The first, I hope you're convinced that lymphoma vaccination improves disease-free survival following chemotherapy in patients who were already in complete remission at the time of vaccination. Again, this concept of secondary prevention or prevention of relapse in minimal disease. The clinical effect of the vaccine was validated by the subgroup analysis of patients expressing the IgM isotype, and this should guide further clinical trials. I didn't spend much time on this, but the long-term clinical experience with the vaccine demonstrates very low toxicity, making it ideal for consolidation or maintenance therapy. And I think this is really where this vaccine is going to have its place in the standard of care, is in maintenance therapy where you need a therapy which is extremely safe. Of course, we have other candidates such as Rituxan and lenalidomide, but as all of you know, these are not entirely safe. And their patients do encounter problems sometimes with maintenance. And finally, as I shared with you, future cancer vaccine strategies should feature combinations with adoptive T cell therapy or immunologic checkpoint blockade, which I haven't had time to talk about. So with that, I just want to show you the most important slide. It was tried to acknowledge their collaborators and contributors as we went along from our own laboratory here, developing all the preclinical studies required for taking that DNA vaccine to a first-in-human clinical trial. And importantly, the Department of Stem Cell Transplant and Cellular Therapy at MD Anderson and our colleagues especially want to thank Dr. Shapal and Ian McNeice because her GMP laboratory is the one that's actually making those myeloma vaccines for the combined trial. So with that, I'll close, and thank you very much.
Okay, we have time for one quick question from Dr. Kwak.
Yeah, excellent talk. Can I just ask, so first of all, apparently you, in lymphoma, you are targeting follicular lymphoma in remission to prevent, to improve the disease-free survival. Actually, why don't you say use diffuse large B-cell lymphoma in remission to do? Have you done that? Actually, you don't see anything, or actually you just restrict yourself to follicular lymphoma? Because I believe in diffuse large B-cell lymphoma, there are more relapse and shorter time interval to relapse. It will be more apparent.
Yes, so the question is about other lymphoma types. And we actually have published on a trial in mantle cell lymphoma with the same principle. We've not done it in diffuse large cell lymphoma, but I think it's a great suggestion. It's just limited by resources of commercial partners.
Definitely.
But I think that's certainly the same principle should apply. And
can I follow on with just one question? The other one is immune evasion by tumor cells has been known for decades. For example, the repression of co-stimulatory molecules that you have been trying to target, CD28. So, so I just wonder, in the success of your idiotype vaccine in follicular lymphoma, what's the mechanism? Why does it not evade by the tumor?
Well, I think we've done mechanism studies from the phase 3 trial, and it appears to be T-cell mediated response. So CD8 positive T cells. So what that means is that the target is on the surface of the tumor cell as a peptide in a groove of an HLA molecule. And I think the future, though, is, as you suggest, to combine the vaccine with checkpoint blockade, such as ipilimumab. I think those are going to be the new adjuvants. And
the last is a very brief question. It's about you mentioned about idiotype vaccine, and the second one you mentioned about fusion of a chemokine with a DNA. Am I correct? So why is the DNA specific to the tumor? It should be a tumor-specific thing.
Yes, I'm sorry.
What part of the DNA is that?
Yeah, so it can be any antigen. It could be any antigen. In fact, that's a platform for vaccine delivery of any antigen, including infectious diseases. But what we're using in the Waldenstrom's trial is the idiotype again.
Okay, great. Thank you, Larry. It's a very nice presentation.